Hey everyone, thanks so much for coming out and joining us for the Psychiatric Neuroimaging Methods and Applications Symposium. We have a wonderful collection of presentations today for some truly amazing junior scientists, so we're all happy that you can make it here. Just for folks that this might be your first day at OHBM, you can ask questions on the OHBM app. And so we'll be taking questions through the application, so there's no in-person questions. You can also upvote your favorite questions if you want. And Elvisha and I can help curate that for the presenters when they finish their talks. So we have a great selection of presentations. First up is Herberto Dhanis, who's not unfortunately able to make it here in person, so this will be an online presentation. But Herberto's up first. Thank you guys.
Hi everyone, my name is Herberto Dhanis, and I'm a PhD candidate at both Olaf Blanke and Dimitri van de Ville's labs in EPFL Switzerland. Before I start, I would just like to say that I would have loved to be there with you in person. Unfortunately, I've had long COVID for the past 4 months, and this has stopped me from traveling and being there with you in person in Glasgow. With that said, it's still a pleasure to be here presenting at OHBM 2022, and today I hope that I can show you how we've used co-activation pattern-based neurofeedback to have participants bidirectionally regulate an hallucination network. So to start off, we're going to be talking about an hallucination called the presence hallucination, which is the very strange sensation of having someone behind you when no one's actually there. And of its many characteristics, probably the most important now is that it's related to sensory-motor processing. We also wanted to quickly allude to its clinical relevance. It's present in many neurological diseases, and for example, in Parkinson's, where patients suffer up to 70% of hallucinations. The early onset of hallucinations like the presence hallucination is a predictor for worse functional outcomes. So our lab has actually made significant progress in understanding this hallucination. We have robotic setups to induce this both outside but also inside the MRI. Here are participants doing poking-like movements in the front, and they are replicated in the back, either in synchrony, so in real time, or with a small delay. And it's with this small delay that the presence hallucination is typically induced. My colleagues Bernasconi and Blondiau also showed that the posterior superior temporal sulcus and the inferior frontal gyrus is more active in the condition where pH is induced versus the condition where it's not induced in healthy individuals. And because this was a clinical study, they also related this to neurological patients with pH. Now, because this was already a very large study, we decided to later on do a follow-up analysis to look a bit more into the neurocorrelates of induced presence hallucination. And we used co-activation pattern analysis to see which regions are co-fluctuating with these two. And so we did this during task, not during rest. And as you know, with CAPS, you get a series of patterns that are activating over time. Once we had that, we decided to model the temporal properties of these co-activation patterns based on condition, CAP, sensitivity to presence hallucination. We did this for occurrences, average duration, and transition probabilities, by the way. And what we found was that in the asynchronous condition where the presence hallucination is induced, and for participants that are sensitive to it, we see a shift in the transition probabilities of all the co-activation patterns to favor a single co-activation pattern that we're calling now the presence hallucination network. This was not observed if you were not sensitive to pH or in any other condition. So with this established, we had now a new objective that was basically to have real-time regulation of the presence hallucination network. Because if this was possible, it would establish a more causal role between the mechanisms and the hallucination, but also answer the question, well, can you, by regulating the network, modulate the hallucination? And to do this, we turned to real-time fMRI neurofeedback, which is based on the premise that if you give informative feedback on brain activity to a participant, they can then search for a mental strategy to actually regulate that brain activity. So to do this, we had a 5-day experimental paradigm where on the first day, participants would come and we would do pH induction in the MRI, and we would also assess their sensitivity to this induction, followed by 3 days of neurofeedback training, and then another day to assess again if there were any changes in pH induction. We had 20 participants doing this, by the way. Starting with the neurofeedback training, we had in each day 3 runs of neurofeedback followed by a transfer run. The neurofeedback runs were composed by blocks of rest, control, and upregulation. A few important things on that. So in rest, participants were really resting, not doing anything. It was in the control that they would start moving the robot in the non-pH inducing condition, and they would listen to the minimum level of auditory feedback independently of what was going on, and they had instructions to try to clear their mind. When we switched to upregulation, the robot would start in the pH-inducing condition, and participants were instructed to have a mental strategy to try to achieve the highest feedback. Now, the feedback here was variable and had an adaptive baseline. What this means is that at all times we were computing a correlational score between ongoing brain activity and the presence hallucination network. But this was always normalized by whatever had happened during the control condition. So the feedback had this adaptive baseline from the previous control block. And then we assessed success of a full neurofeedback run based on a non-parametric procedure that basically looked at the effect size you had achieved between upregulation and control. I'm happy to take questions on that later. You're probably wondering, though, the most important part, what distinguished successful from unsuccessful regulation of the presence hallucination network. And with this, we start on the results. The first thing we had to do to answer that question was to run co-activation pattern analysis again and recover these caps. The most commonly recovered one was actually the presence hallucination cap, which overlapped perfectly with the presence hallucination network. And this was recovered independently. Now, I want to highlight a few other caps we recovered. One of them was the task cap that was more present during both control and upregulation, an anti-task cap, and an anti-DMN cap. Of course, there were a few others. The thing about these two, for example, is that they have overlapping activations with the presence hallucination, especially on the right. And the task cap actually has overlapping deactivations, again, especially on the right, with the activations of the presence hallucination network. And this is going to be very relevant for our results in a second. So participants had basically two strategies for successful regulation, one upregulation and one downregulation strategy. Before I start, I want to quickly say that you always have the models here on top. And again, we looked at transition probabilities, TP, occurrences, or average durations. So our first upregulation strategy compares obviously successful versus unsuccessful runs. And what we see is that in successful runs, participants learn to shift their transition probabilities from the presence hallucination cap to the two co-activation patterns that have overlapping activations with the presence hallucination network. Why? Because this, we assume this gives you a higher feedback simply because the correlation will be higher. We hypothesize that this also leads to a facilitation of then the occurrence of the presence hallucination network because you are already activating regions of this network. And we seem to have this going back to the presence hallucination, especially from the anti-task co-activation pattern. So this is for our first strategy. Our second strategy actually happens during the control condition, and it's more of a down-regulation strategy. So what we observe is that during control, participants significantly recruit less the presence hallucination cap when they are successful, and they also deactivate it. Why? Because we see that there is a higher recruitment of the task cap that I actually had told you had included a partially deactivated presence hallucination network here on the right. And because the feedback is adaptive, you can see how lowering your presence hallucination network during the control will lead to a higher differential that then gives you a higher feedback in the upregulation condition. Also, a very interesting aspect of this is that this is supported by transition probabilities. So you know that you have this inactive state in co-activation pattern analysis, and that you move out of it to have active caps. And what we see is that successful participants learn that when they move out of this inactive state, they preferentially recruit less the presence hallucination cap and more the task cap, which includes the activations over the presence hallucination network in the control condition. So these are our two strategies that participants found. Now, a very interesting question is, do we have lingering effects after this successful neurofeedback training? And I can already tell you that, yes, we do. We had this day 5 and day 1 to assess precisely this. And on day 5, we had a 3-fold increase in presence hallucination sensitivity. How this translated in terms of neurocorrelates, I would say, is even more interesting. Because here we look at differential occurrences between, in this specific case on the presence hallucination cap, between day 5 and day 1. And we see that the more successful you were, the more you activated the presence hallucination cap in the pH-inducing condition, and the less you did it in the non-pH-inducing condition. And these two conditions are comparable to upregulation and to control. Now, the very interesting aspect of this is that it didn't just matter if you were successful or not. If you were, this was actually also regulated by were you sensitive or not to the presence hallucination induction. And only if you became sensitive to this induction do we see this effect. We did not see any effect of success if you didn't become sensitive to pH induction, no matter how successful you were. So this is a very interesting result. And the take-home messages that I want you to have for this presentation are basically that we achieved bidirectional control of the presence hallucination network with an upregulation strategy and a downregulation strategy, and that there were lingering effects that mimicked these two strategies, but only in successful participants that became sensitive to the induction of presence hallucination. So speaking exactly that, you can be as successful as you want, but it has to, you then have to have a behavioral translation for this to linger after neurofeedback training. So with that said, I would like to thank everyone that was involved in the project, especially my PIs Olaf Blanke and Dimitri van der Wiel, without whom this would not have been possible. My collaborators, Giulio, Nathan, Nico, and also Eva and Thomas that were involved in the first project identifying the presence hallucination network for the first time. Again, I'm very sorry that I couldn't be there due to long COVID. Please contact me with any questions you might have, or if you're interested in the project. I have my Twitter and my email here below, and I'll really try to be as responsive as I can. So thanks again for your attention, and enjoy the rest of the conference. Bye.
All right, thanks to Herberto for a wonderful talk. And next up, we have Micah Hetwer. And just a reminder, if you do have questions, make sure to put them in the app in the audience response system. Thanks.
Hi, thanks a lot for having me here today. I'm Micah Hetwer, and I'm super excited to actually share my first PhD project with you, which is on coordinated cortical thickness alterations in mental disorders and a transdiagnostic enigma study. So for me, one fundamental question in understanding the brain basis of psychiatric disorders is to what extent they actually reflect disorder-specific or transdiagnostic features. So I'm here taking the angle of cross-disorder similarities, which I always see as complementary to disorder-specific research. So we know that there are a lot of overlapping risk factors between psychiatric diagnoses, like overlapping genetic risk variants, but also many rather unspecific environmental risk factors, such as early life stress and trauma, which are not super predictive of a certain later diagnosis. And these overlapping risk factors may be one reason for the overlaps we also observe in neuroimaging phenotypes of psychiatric diagnoses, like shared gray matter volume reductions, primarily in the insula and anterior cingulate, as we see in transdiagnostic meta-analyses, but also in previous enigma studies where they look at the spatial overlap of whole brain patterns of disease impact, which they also see to correlate among some disorders at least. So it's another question, what are the features that drive these patterns of illness effects, which we observe to be shared across disorders? And one prominent explanation or theory is that it's linked to underlying network organization. So usually disease effects are not independent and isolated, but rather interrelated across the cortex. But we don't know yet if there's also some kind of transdiagnostic network in which disorder effects covary across the cortex. But this now becomes increasingly possible to investigate through large-scale neuroimaging, thank you, consortia such as the Enigma Consortium, where we can now actually tackle these questions in really large samples. So we here access Cohen's D-maps on cortical thickness alterations, so case-controlled differences in cortical thickness for 6 psychiatric disorders, including data from roughly 12,000 patients and 19,000 controls. And we access this data through the Enigma toolbox, and through that toolbox we also collected healthy connectivity data from the Human Connectome project, so resting state, fMRI, functional connectivity, and structural DTI. And with the aim to find systematic patterns in cross-disorder illness effects, we generated a co-alteration matrix, which you could see as covariance of transdiagnostic illness effects, or mathematically speaking, the correlation of Cohen's d values between regions and across disorders. And based on that matrix, we identified co-alteration hubs by taking the sum of strongest connections. And we see these co-alteration hubs, so regions where disease impact is especially strongly coordinated across the cortex and across disorders, in mainly transmodal regions, primarily the temporal lobe. And we also found these to be not randomly organized across the cortex, but actually correlating with also functional connectivity hubs. So based on that, we next aim to see whether there are also structures that are suspiciously strongly connected to these co-alteration hubs. So we systematically correlated seed-based connectivity profiles from the HCP data with our co-alteration hub map and identified structures that are especially strongly connected to these co-alteration hubs as potential disease epicenters. So we mainly observed epicenters in prefrontal and temporal regions, so heteromodal cortices. And we also see a strong overlap between functional and structural epicenters, which makes sense, I guess, but also functional epicenters emerge beyond structural ones, which can probably be explained by just how functional connectivity is computed, so also including more widespread and polysynaptic networks. So the first chapter of our analysis embeds regional alterations within transdiagnostic cortex-wise covariance networks. And the fact that co-alteration hubs followed the pattern of normative connectivity hubs indicates a certain role of nodal stress in shaping this pattern. Because the idea behind nodal stress is that regions that are strongly interconnected, so active in a lot of different subnetworks and also highly metabolically active, are exposed to more stress and therefore also more vulnerable to pathological changes. So that is a likely explanation for the pattern we observe here. And regarding the epicenters, as said, they are mostly found in heteromodal cortices, so regions that we know to exert mainly long-range connections, so that actually facilitates their contribution to such a transdiagnostic whole brain pattern. And also regions with rather protracted plasticity during neurodevelopment, like the prefrontal cortex, for example, which we of course need for cognitive refinement during development, but also leaves a longer window for aberrant neurodevelopment. In a second chapter of our analysis, we aim to identify cortical axes along which co-alteration patterns were organized using a gradient approach. So gradients are basically just components of a dimensionality reduction, and they reflect the similarity or difference in network embedding in a unidimensional space. Or put simply, plotted on the brain, similar colors indicate similar network embedding. And here we observe that frontal and temporal regions, as you can see on the top right in the principal gradient, frontal and temporal regions have maximally different transdiagnostic covariance profiles. And the second gradient below that tells us that regions holding primary sensory and limbic regions show very different co-alteration network embedding than heteromodal occipitoparietal regions. But we were then wondering, okay, what do these axes actually reflect on a neurobiological level? So we did multiple contextualization steps, two of which I'm showing here, and two others are included on my poster if you're interested further. So we first looked at cytoarchitecture and stratified our gradients according to the von Economo-Koskinas classes, which are cytoarchitectonic classes of microstructural complexity, basically. So we observe that the principal gradient primarily segregates agranular from granular cortices. On the top right, you can see that that is class 1 and 5. So it segregates thick cortex with large cells from thin cortex with many but small cells. So one could say that cytoarchitectonic classes with most different microstructure are also the ones with most distinct illness effect covariance. And the second gradient segregated mainly granular from parietal classes. So overall, we see that areas with shared cytoarchitecture are more likely embedded similarly within pathological networks. Lastly, we also did a functional decoding using the NeuroSense database. For that, we merged the two gradients, as you can see plotted on the colorful brain in the middle. And the colors on the brain also are reflected in the colors of the parcels and their position along the two gradients in a 2D space. So we then also try to associate NeuroSense terms, so functional networks, with their positioning along the two gradients, and saw mostly differential vulnerability in regions involved in basic sensory compared to attention perception related compared to higher level cognitive tasks. So basically the level or complexity of processing is linked to the embedding within these transdiagnostic covariance profiles. Together, our cytoarchitectonic and functional contextualizations imply that susceptibility networks link to regional structure and function. So similar embedding of psychoarchitectonically similar regions may link to different computational strategies or wiring strategies, or also the different degrees of plasticity that are associated with different cytoarchitectonic classes. And we also know that regions with similar microstructural layering profiles are more likely connected. So that could be a certain structural scaffold for the co-alteration network we observe. And similarly, also levels of functional engagement of cortices involved in similar tasks appear to leave brain regions processing similar types of information with shared susceptibility. Lastly, I would like to mention also some limitations of our study, because I always think that's also important. Namely, the disorders we included do have different mean ages of onset, and since we mostly used adult samples, there is likely an offset in disease and medication history. There's also always within disorder heterogeneity, of course, which was not the focus of our study here, but also important to mention. And lastly, we use Enigma data, which is traditionally shared in the Zikin-Kiliani space. So we are left with rather sparse data points and don't have other parcellations that we could replicate our results with. But overall, by looking at regional embedding within transdiagnostic networks, we saw that cortical alterations are coordinated across the cortex and across diagnoses along connectomic, cytoarchitectonic, and functional dimensions. So I'd like to firstly thank you for your attention, but especially my supervisor Sophie Falk and Simon Eickhoff, and the cognitive neurogenetics group and all Enigma working groups involved here. Yeah, thanks a lot.
Sorry. We've got some questions in the app, and one of the top ranked ones are, to which degree can we map these transdiagnostic findings back to the individual disorders?
Thank you for that question. So I would say that different disorders align to different degrees with the transdiagnostic findings. So we actually see that especially disorders with a strong neurodevelopmental component like autism, spectrum disorder, schizophrenia, bipolar match very well to these transdiagnostic findings. And I would also say that most disorders do show hubs and epicenters which overlap to some degree with the transdiagnostic hubs we see. But also when looking at within disorder covariance, so basically coordination across the cortex of illness effects within a disorder, so looking at within disorder covariance compared to transdiagnostic covariance patterns, some disorders are more similar to that, but all of them show similarity in at least some regions. So for example, autism spectrum disorder maps very well to the whole brain pattern we observe in transdiagnostic covariance, whereas OCD and major depression do less so. But it's actually interesting because you do see a distinction that some regions are more similar to transdiagnostic patterns in prefrontal regions and others are more similar in sensory regions. So that also opens up a whole new discussion about transdiagnostics versus disorder-specific understanding, I think.
Great, thank you so much.
Mikey, I think we have time for one more question. So there's a question from the audience on the use of connectivity data from healthy controls to identify disease epicenters. If you could maybe talk a bit about your rationale for using that sample.
Yeah, so there are several reasons actually why we used healthy control connectivity data to identify disease epicenters basically. One being very practical, that we did not have disorder-specific connectivity data, but actually the main reason is rather that we do not expect that in a disorder, all of a sudden two regions that are connected in healthy individuals are just not connected anymore. Of course, there are alterations in connectivity patterns, but they should not be completely different. And also because we use a transdiagnostic approach, it would not have made sense to use like connectivity data from schizophrenia for the whole analysis, or from autism spectrum disorder to look at transdiagnostic epicenters, and there was not a smart way to combine them that I could think of. Yeah, I hope that answers the question.
Wonderful, thanks so much.
Thanks a lot.
Next up, we have an online talk from Xiaosen Zhan. So if you could cue that up, please.
Hi everyone, this is Xiaochen Zhang from the Psychological Health and Imaging Group of Shanghai Mental Health Center, Shanghai Jiao Tong University School of Medicine. I'm very glad to have a chance to present our recent work here, titled Disorder-Specific and Cross-Disorder Brain Morphological Abnormalities Across 4 Mental Disorders. I'd like to thank all the members of the PHI group, especially our principal investigator, Dr. Zhi Yang. Also, I'd like to thank all the co-authors and their research groups for their respective contributions to this work. First, let me give you a background of this research. Current psychiatric diagnoses are based on a categorical system. In contrast, few neuroanatomical alterations that are specific to each diagnostic category have been reported. Therefore, we'd like to fill in this gap and explore the disorder-specific neuroanatomical alterations for 4 major mental disorders. They are bipolar disorder, major depressive disorder, schizophrenia, and obsessive-compulsive disorder. We believe investigations into the disorder-specific alterations likely lead us to a better understanding of disorder-specific etiological processes, and thus to possible specific treatments for different psychiatric disorders. A probable explanation for previous difficulties in searching for disorder-specific neuroanatomical abnormalities is the lack of simultaneous comparisons between multiple diagnostic groups with a constant clinically normal control group. So in this study, we used a single-site multi-diagnosis dataset and directly compared the brain morphological measurements across the 4 diagnostic groups with the same group of clinically normal controls. In addition, investigations into disorder-specific brain alterations call for a framework where the discovered disorder-specific brain alterations could be compatible and not confused with previously discovered transdiagnostic ones. Therefore, we attempted to establish a framework for the operational definitions of the disorder-specific and the transdiagnostic abnormalities to be both compatible and conceptually exclusive to each other. We define the disorder-specific brain alterations as those aberrant from the clinical norm for only one diagnostic group. Particularly, we emphasize that for the other diagnostic groups, such aberrance from the clinical norm should be absent. For instance, this measurement X presented here represents the brain abnormalities specific to disorder C. We define the transdiagnostic brain alterations as those aberrant from the clinical norm for 2 or more diagnostic groups. We further distinguish disorder general alterations from spectrally transdiagnostic ones. The disorder general alterations are defined as the transdiagnostic ones where the multiple groups that show such alterations are similar to each other. For example, this measurement Y, presented here, represents disorder general brain abnormalities shared between disorder C and disorder D. The spectrally transdiagnostic alterations are defined as the transdiagnostic ones where at least two of the multiple diagnostic groups that show such alterations are not similar to each other, but significantly different from each other. For example, this measurement Z presented here represents brain abnormalities that qualitatively shared between disorder C and disorder D, but to significantly different extents. So they may actually be on a transdiagnostic spectrum of certain brain deficits. Based on this framework and on 1,366 cases from a single-site brain imaging database, we extracted 306 regional features, including cortical surface area, folding, thickness, and roughness at 68 cortical regions and the volume of 34 subcortical structures, according to the Descan, Cleany, and Isaac atlas for each subject, with free surfer based on their T1 images. For each feature, the raw values of the direct measurements were transformed into an abnormality value after the age, sex, and total intracranial volume being regressed out via normative modeling, and then were normalized according to the distributions of clinically normal controls. For each subject, the vectorized features formed a profile of brain morphological abnormalities. We first highlighted the direct brain measurements representing disorder-specific and transdiagnostic alterations. Indeed, we found a lot of transdiagnostic alterations in brain morphology in line with previous literature. However, we found disorder-specific alterations only for the schizophrenia group and the OCD group, not for all the four groups considered here. We found that the patients with schizophrenia specifically had smaller bilateral hippocampus than clinically normal controls, and that the patients with OCD specifically had reduced cortical folding at 3 cortical regions. Then, we decided to take a different perspective to look into the data. Considering the high covariances between the direct brain measurements, we further explored the dimensionality of the latent factors underlying the brain morphological alterations across the 4 major mental disorders. We applied an exploratory factor analysis on the individual regional estimates of morphological alterations, and we turned out to reveal a high-dimensional latent factor space. We then identified its dimensions representing disorder-specific and transdiagnostic latent factors that affected brain morphological alterations. This time, we managed to find disorder-specific latent factors affecting brain morphological alterations for each of the 4 diagnostic groups considered here, as well as many transdiagnostic latent factors, which is not surprising. Notably, for bipolar disorder, major depressive disorder, and schizophrenia groups, there is only 1 disorder-specific latent factor for each group, suggesting that all the disorder-specific data covariances across various brain morphological measurements were in a highly similar pattern and thus were organized into a single latent factor that possibly represents a disorder-specific aspect of the underlying etiological processes. In addition, we observed that the individual abnormality profiles in the direct measurement space were mutually more resembling across participants only within the bipolar and the schizophrenia groups, not within all the 4 groups. However, after the space transform, the individual abnormality profiles in the latent factor space were mutually more resembling across participants within all the 4 diagnostic groups considered here. These results support the rationality of the current category-based diagnostic labeling from a neuroanatomical perspective. Interestingly, we found significant topographic correlations between the serotonin receptor density across the brain and the loadings of a particular type of brain morphological measurements to the first three factors, suggesting neurochemical associations of some dimensions of the latent factor space underlying the brain morphological abnormalities. In conclusion, in this study, we attempted to clarify the operational definition of disorder-specific brain alterations. And based on a single-site, multi-diagnosis dataset, we discovered disorder-specific latent factors affecting brain morphological abnormalities for all the 4 major mental disorders under consideration in this study, and disentangled them from transdiagnostic latent factors. We hope the current study may provide novel insights into the neurobiological substrate underlying major psychiatric disorders, as well as into the categorical versus dimensional debate on the intrinsic relationships between various psychiatric disorders. At last, I'd like to thank all the audience. Thank you very much for your listening. Your comments on this work are really welcome. If you have any comments, please feel free to send them by email to the correspondence order. Thank you very much.
Next up, we have Alyssa Day.
I'm just going to test the laser pointer. Oh, okay. If I keep my mask on, is my voice like muffled at all?
It's good.
It's good? Okay, great. Hello, everyone. My name is Alyssa. I'm a master's student, and I'm really excited to share with you my work on linking morphometric patterns of the adolescent psychosis spectrum to different data modalities. Now, psychotic disorders such as schizophrenia are increasingly conceptualized as existing on a continuum that kind of extends from health to illness. And in line with this continuum, subclinical psychotic disorder symptoms also exist at subthreshold, hallmark psychotic disorder symptoms also exist at subthreshold levels in the general population and can be described as psychosis spectrum features. And these features are most common in youth and also increase the risk of subsequent transition to threshold psychopathology in later life. And this gives impetus to understanding the earliest emergence of these features, which my work focuses on. And given that the psychosis spectrum, like many psychiatric disorders, is multifaceted, consideration of its neurodevelopmental, neurobiological, clinical behavioral, and molecular dimensions is warranted. And to this end, magnetic resonance imaging offers the opportunity to capture a neurobiological signature of symptoms by providing indices of different structural measures as shown here. And these features have different maturational timelines and are also thought to reflect different cellular level properties. And so in the context of abnormal neurodevelopment may contribute complementary information. And interestingly, psychosis spectrum individuals have shown changes in individual features that mirror those seen in psychotic patient cohorts. But given the focus on individual measures, it's still unclear whether psychosis spectrum neuroanatomy can be characterized by the coordinated variation of multiple morphometric features, which is a hallmark of maturation. And work on morphometric similarity mapping in the past has shown that by examining covariance across multiple features, we can reveal cortical networks that are more biologically meaningful, such as at the level of within network cytoarchitecture, than single feature-based networks. And while this specific technique is limited by an inability to recover individual feature contributions, more broadly, integrating multiple metrics can improve our ability to identify clinically relevant differences in brain structure. Moreover, previous neuroimaging studies have often compared a single domain or summary score of psychosis spectrum symptoms to brain measures, which does not adequately capture the heterogeneity of the psychosis spectrum. Instead, growing evidence suggests that the psychosis spectrum is underpinned by the simultaneous variation of multiple distinct dimensions of symptoms, each of which represents a continuous phenotypic axis underlying multiple measurable symptoms along which a single individual can vary. And while a consensus model is still missing for the psychosis spectrum, especially in youth, these data-driven dimensions more broadly can better capture variation in neural abnormalities. And to further investigate how morphometric patterns in the PS might confer risk to associated symptom dimensions, we sought to integrate insights from cognition-related functional activation maps, gradients of cortical organization, as well as gene expression of different cell types. And so our first aim was to link morphometric covariance patterns to data-driven symptom dimensions of the PS in youth. And here, using the PNC, which is a community-based sample, we followed previous papers to derive a subsample enriched for psychosis spectrum features. And we then analyzed structural MRI data, as well as psychosis spectrum symptom profiles and sociodemographic features from these youth. Now, first, to characterize multivariate neuroanatomy, we extracted 4 cortical metrics of interest, residualized them for global measures to remove their influence, and submitted this as input to orthonormal projective non-negative matrix factorization. And this method decomposes the 4 metrics into spatial covariance patterns called components, and subject-specific weights onto each component. And the number of components was selected using a stability analysis. And this technique allows us to recover contributions of individual features to components and also identify sparse and non-overlapping components as well. We also ran an exploratory factor analysis to estimate major underlying dimensions of symptoms and computed subject-specific scores onto each latent symptom factor. And finally, to relate subject-specific morphometric component weights to symptom factor scores, as well as demographics, we submitted these 2 data sets to behavioral partial least squares, which identifies maximally covariant patterns of the brain and behavioral data. So our NMF analysis revealed 8 morphometric components, which we kept for further analysis. And looking at the subject level weights, we see that components weren't driven by any single metric. And moreover, neuroanatomically, they capture these largely bilateral and contiguous components, such as the lateral and medial prefrontal cortex, as well as the inferior and anterior middle temporal lobe. Our factor analysis revealed 3 major dimensions of symptoms, which include negative and disorganized symptoms, disordered thoughts or sense of self, and hallucinations and abnormal perceptions. Finally, our behavioral PLS revealed 2 significant latent variables, of which the second was symptom-related, and specifically in this latent variable, older age, male sex, African American race, as well as higher scores on this disordered thought sense of self dimension covaried with cortical changes, including lower gyrification in components 1 and 4, increased and decreased cortical thickness in components 1 and 3 respectively, and lower mean curvature in component 2, as well as lower surface area in component 6. And I'll refer to these as components linked to disordered thoughts or sense of self. So in order to relate these morphometric components to function, to brain function, we first used neurosynth to explore cognitive functional associations of these components. And this reveals that components were correlated with these distinct domains of cognitive functions, which are summarized here. And when we focus on components linked, again, to disordered thoughts or sense of self, we see that they largely correlated with higher order processes such as executive control, emotion, as well as semantic memory. Furthermore, given converging evidence for a large-scale unimodal to transmodal gradient of functional organization, we sought to situate our morphometric components along maturational expressions of this gradient. So here we show component-specific gradient value distributions from the adolescent gradient, and we see that components 3 and 4, as well as, and when we focus on components that were linked, again, to disordered thoughts or sense of self, we see that components 3 and 4 and components 6 and 1 kind of sat along the unimodal and transmodal ends of this gradient, respectively. And furthermore, when we integrate the child and adult expressions of this gradient, we see that components that are initially more unimodal in childhood, such as 2 and 3, shift most dramatically in position along this gradient across maturation, whereas more heteromodal components initially, such as 6 and 1, remain stable in their position, but nonetheless show visible changes in their gradient value distribution. Finally, we asked whether specific cell types might underlie the morphometric components we've suggested to be at higher risk for the psychosis spectrum. And here we use the Allen Human Brain Atlas to extract samples corresponding to each component and submit their sample level gene expression data into a matrix to mean-centered PLS. And this technique identifies gene expression patterns maximally differentiating the input components. And by thresholding the output gene weights, we can obtain PLS positive and negative gene sets, each of which we can then examine for preferential enrichment of genes expressed in one of 7 cell types. So we focus here on LV1, which explained the bulk of the covariance. And in this plot, components separable by the latent variable have opposite signs. And the magnitudes indicate how well they're described by the corresponding gene weight pattern. In other words, their contribution to the contrast. So we see that components 4 and 6 most highly express the PLS positive genes, and component 2 most highly expresses the PLS negative genes. So this latent variable mainly separates components 4 and 6 from component 2. And interestingly, components that were linked to disordered thoughts or sense of self make of all of those that have positive contrast values. And we can further examine the cell type enrichments of the specific gene sets. So in this plot, the circles indicate the true enrichment ratio of a cell type. The box plot indicates null enrichment ratios from random gene lists, and the direction of difference between the two indicates over or under enrichment. So we see that the PLS positive gene set, which is more expressed in the red components, is over-enriched for genes found in astrocytes, microglia, oligodendrocyte precursors, as well as neurons. And similarly, the PLS negative gene set, which is more expressed in the blue components, is enriched for genes found in oligodendrocytes and endothelial cells. So to conclude, we observed that a disordered thoughts or sense of self psychosis spectrum dimension has a distinct signature in these morphometric covariance patterns, that these components converge with large-scale cognitive and functional architectures, which possibly provides a structural functional scaffold for psychosis spectrum emergence, and that components linked to disordered thoughts or sense of self may be coarsely distinguishable from other components by expression levels of specific cell types. Thanks so much for listening, and I'm happy to take questions now or at my poster.
Thanks. So I think in the interest of time, we're just going to do one question. And there's a few asking about why you specifically chose cortical thickness, surface area, and the measures that you did.
Yeah, that's a good question. The first reason was like a practical one. The structural imaging the structural processing pipeline, we used Civet as well as many others, For those pipelines, these 4, cortical thickness, surface area, gyrification, and mean curvature, are really easy to extract. And so it's kind of easy to reproduce. And also these 4 have kind of been shown to index cellular properties and underlying biological properties that are both complementary, but also kind of relate to each other. So for example, folding and surface area are pretty highly correlated, but are thought to reflect different cellular properties. And so we thought that this was a good way to get kind of an accurate picture of structural changes in the cortex while showing kind of the advantage of having multiple features as opposed to just one.
Great, thank you so much.
So next up, we have Sin Kin.
Hello, my name is Sin Kim, and I'm going to talk about my search for associations between polygenic risk for various major psychotic disorders and early adolescent white matter development. And if you're looking at the app, you might have noticed that the title have changed slightly, and I will tell a story about that later. Okay, so here's the big picture behind why I pursued this search. So it is probably well known that adolescence is a very important period for psychiatric disorders because psychiatric disorders are known to be the most common emergence period for psychiatric disorders turns out to be adolescence. And in addition, psychiatric disorders are associated with various white matter defects as large studies from consortiums like Enigma has found. And to bring that together, it is known that adolescent white matter development continues during adolescence. White matter development continues during adolescence. And interestingly, while gray matter development is more linear, white matter development is more nonlinear, and meaning there's a change in slope at around age 10. And I wish to, and my goal was to find out what features predicted how the adolescent trajectories will change at this time point. And yes, so this is my general goal. My general goal is to find predictive factors of those trajectory changes. And for this study in particular, I looked at the baseline white matter and genetics for reasons I will elaborate more. So genetics, I became interested in genetics because psychiatric disorders are strikingly heritable, as seen by many twin and pedigree studies. And also, white matter structures are highly heritable as well. But genes aren't genes expressed at childhood, adolescence, and adulthood and later age aren't always the same. Some genetic effects may be specific to specific age ranges, and for that reason, I thought that some genetic effects may be specific to adolescence, and maybe those changes lead to that emergence in psychiatric disorders. Thus, I hypothesized that genetic risk for psychiatric disorders alters adolescent white matter development trajectories. To investigate this hypothesis, I performed a large data study with the ABCD data, mainly using the ABCD data set, which is a large longitudinal data set, multimodal data set, including genetic DTI, fMRI, MRI, and various behavioral and psychiatric assessments. And based on Based on GWAS results from even larger studies, such as the Psychiatric Genomics Consortium and the UK Biobank, I constructed polygenic risk scores, which are measures of genetic risks for these 5 psychiatric disorders I was interested in. And then I performed a three-way association studies between these three features using linear mixed effects models. And my hope was that based on those results, I would be able to conduct three-way mediation analyses and moderation analyses. But before I get into the interesting story about why my title might have changed, I'm going to go on a slight tangent about my sample size. The ABCD dataset is a huge dataset of 11,000 adolescents. However, only selecting for longitudinal data, only selecting for subjects with available longitudinal data reduced that to around half. And in addition, I had to make a difficult choice that to only select those of majority European ancestry to match the GWAS studies that I was using. And this is because several studies, for example, in a recent study by Schultz et al., they have found that finding polygenic risk score with GWAS studies from the same ancestry has very high replicability and robustness, but cross ancestry, the robustness decreases drastically. And I hope we can only hope that there will be more diverse GWAS studies in the futures to perform similar kind of studies in. And in addition, this is something that I wish I thought about earlier, but I did not. And so when using polygenic risk scores to do association studies, it is very important to control for something called population structure, which reflects difference, not only in wide categories of ancestries like European, African, Asian, and things like that, about population structures within those large samples as well, because those have strong effects tend to have strong effects on the risk score. In addition, although many studies using polygenic risk to investigate behavior phenotypes use socioeconomics as a covariate, I decided not to use them because some recent studies have found that some recent reviews have suggested that socioeconomic variables, depending on how the causal graph is drawn, may be colliders rather than proper confounds. However, including them did not change my results. I also excluded DTI confounds such as head motion and intracranial volume based on the literature. And here are admittedly disappointing results. First results are actually more interesting. I found that polygenic risks were associated with symptom scales, even though the GWAS studies were taken from adult populations. They replicated well in most disorders for adolescents, early adolescents at age 9 and 10. And even more interestingly, I found that the change in symptom was related to polygenic risk in anxiety and depression, which are also strongly correlated anyway, but not in all disorders. For example, ADHD has a strong correlation at baseline, but not longitudinally. And my hope was to find white matter microstructure that mediates that longitudinal effect. However, although when I first submitted my abstract, I found some effects in the mean diffusivity when controlling for the population structure and other compounds based on the causal graph. I did not find. I found fairly robustly that there were no white matter structures that were associated that mediated that polygenic effect. In addition, white matter structure was not associated with the symptoms or with the baseline symptoms. But interestingly, although this is not related to genetics, I found a couple white matter features in the subcortical regions that are associated with that are longitudinally associated with change in symptom scales. And although this was not originally planned, I performed a post-hoc moderation analysis to find investigate more interesting ways the polygenic risk may affect changes in symptoms and changes in white matter structure. And although I have not been able to fully find explanations for all of these, I found that the polygenic risk negatively modulates the effect on the baseline to longitudinal white matter change for bipolar and schizophrenia, which was not reflected in the genetic risk. So that was a bit curious results. And for white matter to symptoms, for the polygenic risk was positively modulated for anxiety, while for longitudinal change, I found negative modulation within ADHD, which again is curious because for genetic risk, the ADHD was not associated with the symptom scales, the change in symptom scales. So with those results that left more questions than answers in mind, to conclude, I found that the one result that survived changes in the causal graphs, my choice in confounds, and all sorts of things was that polygenic risk does predict adolescent ADHD, anxiety, and depression. And this result was further replicated in a completely different study that also released very recently by Weinberg et al. And they also found that depression and ADHD polygenic risk well predicted anxiety and ADHD, but bipolar and schizophrenia were not as predictive for adolescents. So with that robust results in mind about the polygenic risk on the symptom scales, another interesting result that I found was that the polygenic risk predicts anxiety and depression emergence during adolescence. But my hypothesis of white matter structure being a mediator of that was fairly well denied. So I plan to investigate more factors such as the volumetric cortical thickness, functional connectivity, and maybe add more consider more environmental factors in a later study. Another interesting possible limitation that might have prevented me from finding interesting results is that most longitudinal methods require time points of 3 or more. However, the ABCD data set has only collected 2 time points which limited me to using change scores as my longitudinal method. Also, the most significant white matter growth actually happens at around age 14 and 16, which is slightly after the longitudinal time points that I was looking at. And that is the end of my presentation. And if you would like to discuss more about the future directions of investigating how the polygenic risk mediates, what kind of neurobiological mechanism mediates the polygenic risk effect on the change in symptoms, you can come see me at my poster. Also, there are more supplementary results and full code at this link.
I think in the interest of time, we're going to have to skip questions, but please stick around afterwards if you do have any, or check out the poster. And next up, we have Niels Winter.
Hi. So I brought my timer, but I guess the quicker I am, the better. So my name is Niels Winter, and I'm waiting for my presentation for a second. Okay. So my name is Niels Winter. I'm a PhD student at the University of Münster at the Institute for Translational Psychiatry. It's my pleasure to present to you my research on rethinking univariate and multivariate biomarker for major depression. So as you all know, major depression is one of the most common or the most common mental disorder. And although there is a tremendous amount of suffering among the patients, the clinical reality hasn't really changed in the last couple of decades. So many of us are trying to first individualize therapeutic interventions, second, support differential diagnoses, and third, optimize prevention and patient management. And as you may also see, all of these three aspects have one thing in common, and that is we need some form of prediction or classification. And that is why many argue that if we move from group-level inference to a predictive modeling framework, we might actually gain in clinical utility. But let's start first with the more traditional approach and the univariate statistical analysis. So what we have seen in the last years is that it's not that easy to find replicable and reliable differences of brain alterations for major depression. So what we see in the meta-analyses is that sometimes you don't find any significant effects, and sometimes if you pool data modalities, for instance, then you can find something that is significant. The problem is that the effect sizes tend to be rather small. And this is also elegantly shown in the Enigma study from 2016, where they looked at subcortical volume differences. So what they could find is they do find a significant difference of the hippocampal volume, but if you look at the effect size that they find, and this is the largest effect and one of the largest MDD studies, the effect size is 0.17 of Cohen's d. And if you recompute that to a classification accuracy in a single subject framework, and this is what Fried and Kiewit have done, then you actually see that the classification accuracy that you get from this effect is roughly 52%. And you also see this large distributional overlap there. So the question is, the clinical utility of that might not be that high. Okay, but many would argue that univariate analyses probably don't have enough information to lead to single subject prediction. This is why many look at multivariate approaches, and there are many studies out there using machine learning, trying to differentiate between patients or predict some treatment outcome, for instance. The problem with that is that in the last years, and if you look at the reviews, you see a negative correlation between the sample size and the accuracy that you see. So the larger the sample, the lower the accuracies. So this might indicate that there is some form of overfitting happening, and then that the results that we have seen in the last years, 70% accuracy, 80%, 90%, are actually overestimated. And this is why we asked ourselves, if we look at a single study, not a meta-analysis setting where we have a number of different methodological problems and heterogeneity, if we look at a single study, what is the univariate and multivariate classification accuracy, single subject prediction, that we can hope for in a range of neuroimaging modalities? So the sample that we are using is based in Münster and Marburg in Germany. Again, it's a single study with 2 sites. We have roughly 800 lifetime depressive patients. And as you notice, I call them lifetime depressive patients because they're not necessarily acutely depressed, but they need to have one major depressive episode in their lifetime. And this is also the reason why we also looked at depressive subgroups, so subgroups that are acutely depressed or that are chronically depressed, so that they needed to have multiple episodes in their lives. And then we have a control group of roughly 900 healthy controls. So the neuroimaging modalities that we were looking at are structural MRI, so basically the same that the Enigma Consortium has published in this study that I've shown you. Task-based functional MRI, functional connectivity based on resting state, and also structural connectivity based on DTI. And then for resting state and DTI, we also compute graph network parameters as another additional modality or some kind of feature preprocessing, basically. So here you can see the general design of our analysis. On the top of the figure, you see this univariate classification pipeline. And the idea was we take one single modality, for instance, the free surfer parcellation, and then we go through every brain region, as you would do in a mass univariate statistical analysis, and you try to find the single variable, the single brain region that shows the largest group difference between healthy controls and depressive patients. So if you pick that single variable, which is circled in red in this diagram, and then you try to recompute a classification accuracy from that, what do we get? The same goes for the multivariate analysis. So in this case, it's a machine learning analysis. We take exactly the same data modalities, and we feed it through a machine learning pipeline based on our own Photon AI software implementation. And then we basically try to do everything that the machine learning toolbox has to offer. So we do a number of different feature preprocessing steps, dimensionality reduction, feature selection, and then a number of classification algorithms, such as support vector machines, random forests, boosting, and so on. And then for that, all of this is done in a 10x10 nested cross-validation framework. And why nested? Because we also obviously optimize hyperparameters of this pipeline, which is important to get a good classification accuracy. And then in the end, we find one hyperparameter configuration, so a combination of feature selection, for instance, and a classification algorithm that is optimal, and then we check out of sample the classification accuracy. And these are the results. So starting with the univariate results, what you can see on the left is that across the different data modalities, the univariate classification performance that we get, again, in a single variable showing the largest group difference, is between 52 and 55% accuracy, which mimics the results that Fried and Kiewit have shown. What I find even more remarkable and striking is if you look at the distributional overlap, which is in the middle, or if you look at the right, there you can see the resting state connectivity data, which have the strongest effect. And if you look at the distributional overlap, I think it's quite impressive, and it becomes evident quite clearly that a single subject prediction or classification based on this single variable is not possible. If we move forward to the multivariate analysis, then what we can see, and what I find even more surprising, is this does not change. So we do increase a bit in classification accuracy. So we go up to around 60% for resting state again. But the overall effect is way less than we have seen in the last years of publications. This does not change if we look at acutely depressed or chronically depressed patients. And there's one nice thing for the machine learning pipeline, that we can also combine all the different data modalities. So we can put everything into one model and use different methods of data integration, and then see how far can we go if we combine all this information. There are a number of problems associated with that approach, but then we also see we don't exceed the 60% mark. So apparently this is a ceiling that we hit. Okay, so with that, I want to conclude and say that from our results, we see that healthy and depressed individuals are nearly indistinguishable on a single subject level, and that's true across the different neuroimaging modalities. So this is why I titled my talk Rethinking Multivariate and Univariate Biomarker for Depression. But now the most interesting question is, what are possible reasons for that, and where do we go from here? So in the interest of time, I only want to emphasize one particular point that I find especially important, and that is, we also heard that in a couple of talks, depression is not a homogeneous disorder. We have vast clinical but also neurobiological heterogeneity. And this leads me to approaches such as identifying depressive subtypes, which is what we're also trying to do with our data set, or focusing rather on symptoms than on syndromes, which reduces the clinical heterogeneity. But again, it's questionable whether we can find better classification accuracies in the end using just a smaller effect if you only look at one symptom. Okay, and with that, I want to thank you all for your attention, and I'm very happy to discuss more of these possible reasons for this effect. And my poster, I think it's in roughly an hour, so yeah, happy to discuss more. Thank you.
So I just want to say a quick thank you to everyone in the audience for your attention and attendance at the symposium. Due to time, we're not going to be able to take any more questions, but I'm sure if you grab the speakers at their posters, they'll be happy to discuss their work. So thanks again, everyone, and have a great lunch.